Hey everyone, and welcome to this explainer. I'm your AI narrator, and I am absolutely thrilled to welcome all you first-year MBBS students tuning in today. We're going to walk through a really simple, super high-yield physiology explainer on the male reproductive system. We've got a lot of fascinating science to cover, and my main goal here is to make this as crystal clear and memorable as possible for your exams. But first things first, it's really important that we establish where all this excellent information comes from. Content by Dr. Neeraj Mahajan and Dr. Kartik Patel, physiologists from the heritage city of Ahmedabad and Gujarat, India. We are incredibly grateful for their structured academic work that makes up our source material for today. Okay, here's a quick look at our agenda for today's session. Number 1, testis temperature. Number 2, spermatogenesis stages. Number 3, testosterone lifespan. And number 4, we'll wrap up with a quick revision. Right, let's dive into Section 1, testis temperature and anatomy. To really understand the unique environment required for male reproduction, we kind of have to start by asking a really fundamental anatomical question. Why is the testis located down in the scrotum, entirely outside of the main abdominal cavity? I mean, it seems a bit vulnerable out there, doesn't it? Well, physiology has a pretty brilliant reason for this design. The simple answer? Temperature. You see, making sperm, a process we call spermatogenesis, actually requires a temperature that's considerably lower than the deep, warm interior of your body. Specifically, the testes need to be kept at a nice, cool 32 degrees Celsius, which is about 2 degrees lower than your normal body temperature. This cooling is maintained partly by air just circulating around the scrotum, but more importantly, it relies on this amazing countercurrent heat exchange system between the spermatic arteries and veins. Basically, the warmer blood coming in is cooled down by the cooler blood leaving. It's like a built-in radiator. Because if that temperature gets too high, well, sperm simply cannot be produced. Moving right along to Section 2, spermatogenesis stages, the cellular factories. So there are 4 chronological stages that transform a primitive, early germ cell into a fully mature spermatozoon. These steps are proliferation, growth, maturation, and transformation. Let's break them down. It all kicks off with proliferation. Primitive cells called spermatogonia basically multiply by mitotic cell division. They're just copying themselves over and over again inside the testis. Then, in the stage of growth, they cross a special barrier and literally just grow larger in size, becoming what we call primary spermatocytes. Next up is the stage of maturation. And honestly, this is where the real magic happens. This is meiotic division. So the primary spermatocyte divides into 2 secondary spermatocytes. Then, those secondary spermatocytes divide yet again to form 4 spermatids. This double division is exactly how the body cuts the genetic material right in half. Now, this brings us to absolutely crucial point, the chromosome count. Pay attention because this is super high yield for your exams. The primary spermatocyte starts out with 46 chromosomes, so it's a diploid cell. But after that first meiotic division, the secondary spermatocyte only has 23 chromosomes. And those final spermatids, they also have 23 chromosomes. They're haploid cells. This is essential, guys, because the sperm has to combine its 23 chromosomes, but the ovum's 23 chromosomes to make a human with 46. Finally, we reach the stage of transformation, which is also known as spermiogenesis. Notice that the spermatids don't divide anymore here. Instead, they just physically reshape themselves. They shed a ton of extra cytoplasm, they pack all their genetic materials super tightly into a head, and they grow a long tail. They completely metamorphose into those mature sperm cells you're probably familiar with. All right, Section 3, testosterone lifespan and hormone functions. So testosterone is secreted by the interstitial cells of Leydig inside the testes. But here's the thing. It doesn't just do one single job. It actually has completely different roles depending on what stage of life a male is in. We can break this entire timeline down into 3 distinct phases: embryonic life, puberty, and adult life. Let's keep it very simple. Before a baby boy is even born, during embryonic life, testosterone is already hard at work. Its main jobs here are the differentiation of the male's sex organs, basically ensuring the body develops male anatomy instead of female. And, importantly, it stimulates the descent of the testes from up inside the abdomen all the way down into the scrotum. Now fast forward to puberty and adulthood, and testosterone triggers some massive systemic changes. It causes a huge increase in muscle mass and thicker, stronger bones, and that includes developing a funnel-shaped pelvis. It also deeply affects the central nervous system,
influencing behaviors like aggression. And it enlarges the larynx, or the voice box, which is exactly what makes a man's voice drop and become much deeper.
Oh, and of course, testosterone totally dictates hair distribution. It triggers hair growth on the face, the chest, and the body. But ironically, while it causes hair to grow almost everywhere else, it's also the exact same hormone responsible for decreasing hair on the top of the head, ultimately leading to male pattern baldness. Kind of a crazy trade-off, right?
Okay, Section 4. Quick revision to test your knowledge. Huge congratulations on making it through the core content. Now, let's do a really quick active recall session just to lock all this in. I want you to try and answer these in your head before I reveal the answer. Ready? Question 1. What is the normal temperature required for spermatogenesis to occur successfully? I'll give you a second to think. Got it? The answer is 32 degrees Celsius. Remember, this is about 2 degrees cooler than your normal body temperature, and it's maintained by the scrotum's location and that brilliant counter-current heat exchange system we talked about. Great job if you remembered that.
Let's try question 2. This one focuses on those really high-yield division facts. How many chromosomes does a primary spermatocyte have compared to a spermatid?
Right. So the primary spermatocyte has 46 chromosomes, while the spermatid has exactly half of that, at 23 chromosomes. That is the vital shift from a diploid cell to a haploid cell, all thanks to meiotic division.
Okay, here is our third and final revision question. We literally just covered this timeline. What are the two main functions of testosterone during embryonic or fetal development? And the answers are, number 1, the differentiation of the male sex organs, and number 2, the descent of the testes down into the scrotum. You have worked so hard today, and honestly, absorbing this complex physiology is no small feat. You should definitely be proud of yourselves.
Before we wrap up, I want to leave you as a final thought to really ponder. How are you going to apply this foundational physiology in your future clinical rotations? Think about it. If a baby is born with cryptorchidism, which is an undescended testis, how might being stuck up in that higher abdominal temperature impact everything you just learned about spermatogenesis today? Keep asking yourselves these big questions, keep connecting the dots, and keep learning. Thank you so much for joining me on this explainer today, and I'll see you next time.